Over an aerial view of the River Tay and the Life Sciences building, text reads, University of Dundee, School of Life Sciences. T-cell growth and division. Microscopic footage shows a T-cell. Text, T-cell in action. Footage courtesy of Ritter Lab. An orange cell attaches to a blue cell.
If you have recovered from a cold recently, you should probably thank your T-cells. Your body has an amazing defense against infections called the immune system, which is made up of many types of immune cells. We study one type of immune cell called a T-cell. We are interested in how they grow and increase in number.
A mass spectrometer.
We use a scientific instrument called a mass spectrometer, or mass spec, to study them.
A scientist removes a 6-well plate from a fridge. Some of the wells contain a pink-tinged liquid. She pipettes liquid from a tube and injects it into the liquid in the wells.
The first step is to create an immune response. We do this by activating the immune cells with antibodies.
She places the plate under a microscope and examines it. A grey, microscopic image shows T-cells.
We are specifically interested in items called proteins that are made by the T-cell during the immune response.
The scientist pipettes detergent into a sample tube and then places the tube into a centrifuge. She then studies the tubes. Sediment has gathered at the bottom of both tubes.
To get to them, we break open the T-cells by adding detergent. We collect the protein and wash it several times.
She adds an enzyme to one of the tubes, then taps it with her finger. She places the two tubes into a thermal mixer.
Next, we add an enzyme to chop the proteins up to make them smaller. Then we put it in a thermal mixer. This machine heats and mixes the contents. We call what we will study in the mass spec the sample.
The scientist then adds the sample into a pipette tip with a white filter inside that is held in each of the tubes and returns them to the centrifuge. She examines a tube, where the liquid has gathered at the bottom of the tube.
The sample is not quite ready to go into the mass spec yet. The mass spec is extremely sensitive to salt, so we need to remove salt from the sample.
The scientist carries the samples into a room where another scientist works. The other scientist places the samples into the mass spectrometer. As the machine operates, they discuss data displayed on a computer. Later, the second scientist sits in an office and reviews data on another computer.
We are now ready to place the sample into the mass spec. After 3 to 4 hours, a single mass spec run gives information on 4,000 to 6,000 proteins. This includes the type and amount of each protein in the sample. This data is a large fraction of the proteins present in the cell. We now need to study the data collected from the mass spec. We spend a lot of time analysing the data using Excel and approaches that allow us to handle big data.
A graph is displayed on a laptop. Elsewhere, the two scientists sit at a table and talk with two others. Data is displayed on an open laptop on the table.
These data help us to better understand how these T immune cells work. They are crucial for our body's fight against viruses and cancer cells. But if these T cells become too active, then it can result in autoimmunity. So, it is important to research how these immune cells act, including how they grow and increase in number.
A black letter W appears over text to form the Wellcome logo. Text below reads, supported by the Wellcome Institutional Strategic Support Fund awarded to University of Dundee. A blue and white crest appears beside blue text to form the University of Dundee logo.